Okay, now you need to remember that complete dominance, where there's one dominant allele and one recessive allele, is only one of three types of dominance. We also have incomplete dominance and codominance. Now, incomplete dominance is where no allele is dominant over the other. Okay, so there's no recessive or dominant allele. When they are crossbred, they produce a third characteristic in the offspring. For example, if a flower with white petals was crossed with a flower with red petals, the offspring would have a combination of the two, a mixture, and it would then have pink petals. Codominance is when both alleles are equally dominant. So with the same example, if you had a red flower crossed with a white flower, the offspring would have a combination, an equal amount of white and red on its petals. That is codominance. Okay, now it's quite easy to represent these with a genetic cross. They are almost identical. If you look at this page, please, you see I've made some additions in red pen to your textbook. All right, please make sure you set it out in the way I've done it here. Okay, incomplete dominance was where you're combining red flowers with white flowers, and the resultant offspring will have pink flowers. So please look at the key. Red, we will have two capital R's representing red petals and two capital W's representing white petals. And then also in our key, we have to include the pink petaled offspring, which will be a capital R and a capital W. Okay, so once again, we set it out. We know it's Pip got me greedy for fig pie. Okay, phenotype is red times white. We represent the alleles in the genotype, capital double R times capital W, double W. Meiosis, we write nothing next to it. Then your gametes is your R, or R comma R times Y comma Y. You would then do your Punnett square just to help you work out what the offspring would, what the alleles would look like. Okay, you would then with your genotype have 4 RWs. That would mean 4 pink petaled offspring. Okay, codominance, again, we have our key, double capital R for red, double W for white, and then RW is now expressed not as pink, but as partly red, partly white. Okay, this is the only real difference between these two genetic crosses. Your genotype, red times white, phenotype, sorry, in words is red times white. Genotype, capital R, double R times capital double W. Meiosis, and then your gametes is R comma R times W comma W. Fusion, we write nothing next to. Your genotype, you will have 4 capital R's, capital W's. Phenotype, you will have 4 flowers that have an equal distribution of red and white. Okay, not pink. That is the only difference, the expression in the phenotype. Okay, for codominance, equal. For incomplete dominance, a third characteristic, pink. We are now going to do activity 5.4. Please do it with me. Okay, that is on page 77. Right, number 1. In a certain species of rose plants, there is incomplete dominance between the alleles for red and white flower color. This leads to the production of a third phenotype of pink flowers as an intermediate characteristic. Okay, so they've already told us this is an incomplete dominance genetic cross. They've told us that there's a third characteristic that comes out in the offspring. So now the question is, determine the genotypes and phenotypes of the first filial generation if a red flowering rose plant is crossed with a white flowering rose plant. Right. So that is 1.1. We will have a key first. Capital double R for red, double W for white, RW for pink, and then we set it out. Pip got me greedy for fig pie. Okay, so phenotype we describe in words first. Red crossed with white. Genotype is your alleles. There's no dominance here. We write them both with capital letters. We leave open meiosis. The gametes, we split these alleles here with a comma. Fusion we leave open. We do our Punnett square. Okay, our R will come first. And you will notice that all 4 combinations gives rise to capital R, capital W. We will write this in the genotype. Our phenotype is then 4 pink, according to our code, our key, 4 pink flowered offspring. Right. Now we will do 1.2. It tells us, determine the genotypes and phenotypes of the second filial generation if the offspring in F1 were interbred. Okay, so that's the offspring. We're going to interbreed 2 of them. We're going to interbreed 2 pink flowers, and we are going to see what the results are from there.